Hey everyone, and welcome back to Smart Tutor. Before we get started, if you enjoy our deep dives, make sure to hit that like button and subscribe so you don't miss a thing. Now I want you to imagine a war, but it's not being fought on some distant battlefield. It's happening on a microscopic scale deep inside the human body. The enemy? It's relentless, a rogue version of our very own cells. And the weapons we use are powerful, incredibly sophisticated drugs designed for one single purpose: to seek and destroy. So today, let's dive right into this battle and really understand the core strategy behind one of its most powerful tools: chemotherapy.
First things first, in any war, you have to know your enemy. So on one side, you've got your normal, healthy cells. They look pretty organized, right? They've got a clear structure, they follow the rules, they do their job. But then, you look at the other side. That's the cancer cell. It's just chaotic, kind of misshapen, and totally out of control. The difference is just, wow, it's night and day. And it's this very chaos that actually gives us a target.
So let's zoom in on those differences. A normal cell has a lot of this stuff called cytoplasm and a single, neat little nucleus with fine chromatin. But cancer cells, they flip the script. They have a small amount of cytoplasm, but their nucleus is all over the place. Often there's more than one, they're huge, irregularly shaped, and the chromatin inside is coarse and all clumped up. Now, this isn't just about looks. It's a huge red flag that the cell's command center, its very DNA, is fundamentally broken.
So what do these broken cells actually do? Well, their one and only mission is to multiply, and they do it with terrifying speed through a process called mitosis, but it's completely uncontrolled. They are basically in a constant state of building, what we call anabolic activity, while their breakdown or catabolic processes are turned way down. They're just rapidly churning out the materials to build more of their corrupted command centers. Think of them as tiny, rogue factories working overtime to clone themselves, totally ignoring all the body's signals to just stop. But here's the twist. This reckless speed, this obsession with division, it's not just their greatest strength, it's also their single biggest weakness.
And that brings us right to the heart of the chemotherapy strategy. I mean, if cancer's main weakness is its constant non-stop division, well, the plan is simple: attack that exact process. To do that, we need to map out the battlefield where all this division is happening. We have to know exactly when and where to strike to get the most bang for our buck. And this is our map. It's called the cell cycle. You can think of it as the life story of a single cell, from the moment it's born to the moment it divides into two new cells. It all starts with G1, which is a growth phase. Then it hits the really critical S phase. That's where it synthesizes or copies its entire set of DNA. After one last growth check in the G2 phase, it finally enters the M phase, mitosis, and that's where it physically splits. This cycle, this is our battlefield. Now, on this battlefield, we really have two fundamental attack plans. The first is what we call cell cycle specific, or CCS for short. These are like highly trained snipers. They only work during a very particular phase of the cell cycle, say right when the cell is busy copying its DNA. The second plan is cell cycle nonspecific, or CCNS. These are more like heavy artillery. They can damage a cancer cell at pretty much any point in its life, whether it's actively dividing or just kind of hanging out and resting.
And this really shows you how we deploy our forces. Over on the left, you've got the non-specific agents, our heavy artillery like alkylating agents that can strike anytime. And on the right, you have our specific agents, our snipers, that are timed to hit these really vulnerable points. The anti-metabolites go for the S phase, the vinca alkaloids hit the M phase, and so on. Each of these is a different class of weapon, designed for a very specific job. So let's take a closer look at these weapons.
First up, let's talk about that heavy artillery, the cell cycle nonspecific agents. A classic example of this class is something called alkylating agents. These drugs, they don't need to wait for the perfect moment. They just go straight for the enemy's command center, no questions asked. So how do they actually work? Well, think of them as tiny chemical saboteurs. They get inside that cancer cell and just start wrecking its DNA, the cell's fundamental blueprint. They create breaks in the DNA strands, they cause the genetic code to get misread, and once its blueprints are all scrambled, the cell can't function anymore. And most importantly, it can't replicate itself. Game over. So the key here is that these alkylating agents, they just bypass all the other cellular stuff and launch their attack directly on the DNA itself, no matter what phase the cell is in. It's this direct, overwhelming damage that makes them so powerful, and it's why they're effective whether that cell is actively dividing or not.
All right, let's switch gears and talk about our other weapons, the cell cycle-specific agents. Now, these guys require much more precise timing. Our first example is a class of drugs called anti-metabolites, and they are absolute masters of deception.
Okay, so these drugs are basically spies. They're designed to look almost identical to the natural building blocks a cell needs to make new DNA. The cancer cell, in its crazy rush to divide, accidentally grabs these imposters and takes them inside. But once they're in, they just clog up the whole machinery, blocking the key enzymes the cell needs for DNA synthesis. And the timing is perfect. This attack hits right during the S phase, the exact moment the cell is trying to copy all its DNA.
The strategy is brilliant, really. Agents like methotrexate and 5-fluorouracil that get into the system and just mess with the production of purines and pyrimidines, those are the essential building blocks for DNA. By shutting down these supply lines at such a critical moment, they pretty much starve the cell of the materials it absolutely has to have to replicate.
Now for another class of these highly specific agents, and they use a completely different tactic. Let's talk about the vinca alkaloids.
So, if the anti-metabolites sabotage the blueprints, the vinca alkaloids are here to sabotage the assembly line. These agents target the M phase, or mitosis, which is that moment the cell is trying to physically pull itself into two. They do this by stopping the formation of something called the mitotic spindle. You can kind of picture it as a molecular rope and pulley system that pulls the copied chromosomes apart. Without that system, the cell is just frozen in place, totally unable to complete its division.
Okay, so let's pull all this together for a second, because this is the crucial part. We have different weapons for different situations. Think of this as our little mind map. You've got your alkylating agents, which are nonspecific. They hit the DNA anytime. Then you have the anti-metabolites, which are specific. They target DNA production during the S phase. And finally, the vinca alkaloids, also specific, which stop the physical division process in the M phase. Different tools for different parts of the job.
But, waging a war like this, it's not clean. It's not without consequences. Attacking cells, even the cancerous ones, can cause some serious collateral damage. So let's talk for a minute about the aftermath of these treatments.
So what is the cost of this cellular war? Well, it turns out that the very thing that makes chemotherapy so effective against cancer cells, its ability to kill rapidly dividing cells, is also the very thing that causes its most famous side effects.
See, cancer cells aren't the only rapidly dividing cells in our bodies. So are the cells in our bone marrow, in our hair follicles, and in the lining of our stomach and intestines. The problem is, chemotherapy can't always tell the difference between a cancer cell and a healthy, fast-growing cell. This leads to that collateral damage, things like bone marrow suppression, hair loss, and nausea. It's the really difficult trade-off at the heart of this entire treatment.
So how do we manage this? Well, the solution is a combined arms strategy, what we call combination therapy. By using several different drugs at once, we can attack the cancer from multiple angles, which makes it way harder for the cancer cells to adapt and develop resistance. And crucially, it also lets doctors use lower doses of each individual drug. This helps keep the treatment inside a sweet spot called the therapeutic window, strong enough to kill the cancer, but not so strong that the side effects on our healthy cells become totally overwhelming.
And all of this leads us to the new frontier in this war. While traditional chemotherapy is incredibly powerful, the future is really moving towards more precise weapons. What we're looking at here is a monoclonal antibody, which is a type of targeted therapy. Now, instead of just attacking any fast-growing cell, these agents are engineered to lock onto specific proteins that are only found on the surface of cancer cells. It's like a guided missile. And this allows for a much more targeted attack, which minimizes all that collateral damage to healthy cells we were just talking about. So we really move from an era of, let's call it cellular carpet bombing, to one of precision strikes. The evolution of this microscopic war is just a testament to our growing understanding of the enemy within. The strategies are getting smarter, they're getting more targeted, and they're getting more effective. And it really leaves us with a critical question. As our weapons get more and more precise, what will the next great breakthrough in this battle even look like?